Presented by JOYVE. Introducing our first and foremost personal product, 3-in-1 PV Kit Test by JOYVE. Made easy, made simple. PV is one of women's most common infectious diseases that might cause inconveniences in your daily activities. JOYVE is coming up with your one-stop solution in diagnosing PV. With this 3-in-1 PV test kit, made PV easier to be diagnosed earlier even by yourself. Thus, it can be treated faster. It's just what every woman has been waiting for. This is our design story, the idea, the science behind it, research and manufacturing process, and finally, how our product may affect every woman's life. Mucus has been the most promising tool to determine the health of woman's reproductive system. Approximately, a woman produces 2 to 5 milliliters of clear, thick, and odorless vaginal discharge. The changes that occur might lead to speculations, if not accompanied by further confirmation. Evermore, it might be confusing to differentiate the mucus that is being produced by vagina. Woman's reproductive system, especially vagina, is a home for thousands of microflora. Over 120 species of lactobacillus have been identified, and more than 20 species of streptococcus have been detected in the vagina, such as lactobacillus, staphylococcus, streptococcus, Gardnerella vaginalis, and septic. Most of these bacteria will do mutualistic symbiotic relationship until their number grows rapidly and start to cause problems. What are the general symptoms? How do most women identify the presence of PV in their private area? First one is increased vaginal discharge, watery, gray, or white in color. Then, the fish-like odor, burning sensation during urination, unpleasant sensation such as itching or irritation in the vaginal area. Lastly, PV will increase the risk of contracting STDs and other health problems. Then, how our diagnostic kit works, we'll tell you in the science behind it. As its main target to provide you with the most comprehensive and fast diagnostic tests, this kit will be included with 3 main tests, specifically with tests or odor tests, pH tests or acidity, and PV antibody serological tests with several solutions provided along the kit. Most patients with PV will have fish-like odor in their vaginal discharge. To confirm the availability of this odor, we will use 10% potassium hydroxide, which will be mixed with the mucus. The positive indicator is a strong fish-like odor that comes from the release of amines like putrescine, cadaverine, histamine, and trimethylamine. For the acidity test, we will provide you with a reaction. The reaction between the solution and the mucus will have different color appearance depending on its acidity. For normal vaginal condition, the pH is ranged from 3.8 to 4.5. In the presence of the PV, vaginal discharge pH will be affected and increase greater than 4.5. In this condition, male red will change into yellowish color as a positive PV indication. For our third test, PBS as a buffer solution will be added to a woman's vaginal discharge. Using the principle of lateral flow assay in nitrocellulose membranes, bond between antigens like NAMH2 and NAMH3, and our antibodies will be determined the result. We'll explain it further in the next slide. As you can see, we got Gardnerella vaginalis as one of the most dominant pathogen of PV. produces NH2 and NH3, or also known as sialidase, as extracellular enzymes, which are disposed of vaginal mucus and will be placed at the sample pad along with this buffer as a diluting agent. In the presence of NH2 and NH3, the diluted mucus will move along with these enzymes to the next pad by capillary motion. At the conjugated pad, these antigens will meet their own matched antibody with gold nanoparticle conjugated at the antibody's constant fragment. We combine our capture antibody with gold nanoparticle so we could avoid a false positive signal and maximize the amplification of detection signal of the enhanced LFA. At the nitrocellulose membrane, these antigen-antibody probe complexes will meet the capture antibody and form two test lines and a control line. The excess fluid and unnecessary substances will keep moving forward to the absorption pad. There are 5 possible results. 3 colored stripes mean positive for BV, single colored strip at control line means negative for BV, 2 colored stripes at test lines mean invalid result, and lastly 2 colored stripes, a test line and a control line, means further tests is needed to confirm whether the infection is resulting from GV or other pathogens. How do we confirm the presence of BV if the result means further tests are needed? In this case, we need a golden standard for BV detection, called as Nugent's score. The basic principle of this test is to use gram staining and count the presence of disruptive epithelial cells under the microscope. The positive result is when the score reaches above 7. Next, we will tell you how we produce our diagnostic kit. In the first step, we will need to isolate colonies from GV bacteria and culture it in an enriched medium. The NH2 and NH3 gene isolation will be done in the following colonies. Primer for amplification using PCR is designed and amplify the number of copies for NH2 and NH3 genes. These genes will be inserted in an expression plasmid which later will be inserted into a competent E. coli cell. The colony of transformed bacteria will be cultivated and selected using antibiotics as selectable markers. NH2 and NH3 protein production is induced by isopropyl beta-thiogalactopyranoside solution. The protein will be collected after the bacterial cell outlies. NH2 and NH3 protein are confirmed using SDS page. And later, the purification will take place using affinity chromatography, which will give us the extracted target proteins. In the production of antibody candidates, the antigens consisting of purified NH2 and NH3 are injected to mice. Later on, the spleen will be isolated and degraded into single-cell lymphocytes. The lymphocytes will be fused with myeloma cells, producing hybridoma cells. Those cells will produce our antibody candidates in massive amount. Our anti-NH2 and NH3 proteins will be then inserted into LC-MS or MS technique to finally determine its primary structure. With the acknowledgement of the primary structures, we can build a 3D structures using Rosetta antibody and the test of competencies of antibody candidates using Stackdom. With the selected anti-NH2 and NH3, we will do a resuspension of antibodies to gold nanoparticle as per. The result is a gold nanoparticle that is conjugated to the basal region of the antibodies. The goals. How do our product will affect your life, what will be changed? Through this product will show you a game changer and innovation made only for your comprehensive, competitive, and convenient diagnostic care. From Joyce for a better living, thank you.